Hi, I'm Dr. Michael Fossel. I'm going to talk about aging and rejuvenation and specifically about the prospects for intervention. Putting it more bluntly, I'd like to talk about the prospects for reversing human aging at the fundamental cellular, genetic, and epigenetic levels. I find that most people actually don't believe you can intervene with aging, or if they do believe you can intervene in the aging process, they think you can do it with small molecular approaches, that is, attacking basically biomarkers and signs and symptoms of aging, rather than understanding the fundamental process, which is more critical. And I'd like to point out there's a difference between knowledge and understanding. Many people know a great deal about aging. I think many of us do, but that's different than understanding it. And often the problem is that we don't examine our fundamental assumptions about how the aging process works. For example, we tend to assume that it's no more than wear and tear, that is entropy, without considering the complexities involved at the cellular level, conceptually and physically. Let me give you an analogy. This is a quote from Einstein that I think nails it. If I go back 500 years, the Vatican astronomers had a great deal of a great deal of knowledge about planetary positions. They had excellent data, but they had no understanding of orbital mechanics. And it took people like Johannes Kepler, in this case, Copernicus, to explain what was really going on because they had a deeper understanding of the process rather than simply knowing the data. So the difference between knowing and understanding is enormously important. And when it comes to aging, we tend to know a great deal. For example, mitochondrial dysfunction, changes in DNA repair, changes in turnover rate of molecular pools, but that's not the same as understanding how those things work. This is a quote from one of my favorite Chinese poets, Li Bai, some 1,200 years ago. And if you hunt only rabbits, then you don't catch tigers and dragons. And too often with regard to aging and rejuvenation, and in fact, age-related diseases, we tend to go after the rabbits. For example, simply mitochondrial dysfunction or NAD or resveratrol, or without understanding the process involved that lies beneath those things. So I have been intrigued by this entire question now for in excess of 45 years, starting here on this campus where I got my MD and PhD years ago. And what I found is that it's sort of like the elephant in the blind man. When it comes to aging, we tend to know a great deal about individual parts of the elephant, whether it's the ear or the trunk or the tail or the side of the elephant, without understanding the overall elephant itself, that is the entire bony structure and the organism. We tend to understand only parts of the trees and not the forest. And it intrigued me enough that I stayed with this and then gave the first time in history, gave the first talk on reversing human aging here at the NIH 25 years ago this year. And the point I was making was that, again, if we can understand the fundamental process of aging, truly understand it, then the potential is to be able to cure and prevent all age-related diseases. And I think that's something that we probably can do. And I made that argument first, now again, 25 years ago in that book on the left, and in the first articles ever in the medical literature in the Journal of American Medical Association at about the same time, published the first and still only textbook on this with Oxford University Press, and then wrote a more recent book, The Telomerase Revolution, talking about this process again for the general public. But if you look back historically, what we have done for aging is essentially nothing. The mean lifespan has basically tripled in the past 200 years in most countries, but that has not affected the maximum lifespan and the aging process per se. We live longer because we tend to deal with early diseases, for example, microbial infection. And in fact, if I look back historically, the single major medical advance we've made in the last 200 or more years has been a conceptual one, that is understanding a microbial etiology for diseases. And that's allowed us to do things like washing hands before deliveries, which drastically cut the fetal or the neonatal and maternal death rates, the early immunizations, vaccinations, the earliest antibiotics. And this not only improved the length of life, but the quality of life, and actually lowered the cost of medical care. An example, in 1950, there were estimates that most countries would be medically bankrupt because of the cost of treating polio victims, that is iron lungs and rehabilitation, nursing care, pulmonary toilet, that were expensive. But by the year 2000, we were not medically bankrupt because of polio. And in fact, the WHO estimated that per capita cost of treating polio was less than 10 cents per hit. So the advance was not in improving the iron lung, but was improving our understanding of polio in that case and how we could treat and prevent it. I think the same thing is about to happen in a much more drastic sense with regard to age-related diseases. What we are doing is finally beginning to get a grasp on the fundamental processes of aging, which I'll get back to in a minute. And that will allow us to drastically undercut the costs globally, nationally, and personally of treating age-related diseases, and in fact, cure and prevent such diseases. This was first demonstrated, as I will show later, in human cells, basically 23 years ago, and in human tissues about 21 years ago. And we're on the verge of doing this with whole organisms, as has been partially done already in the last 10 years. So one of the problems that we have had, particularly with regard to age-related CNS diseases, that is neurodegenerative diseases, dementias, Alzheimer's in particular, is this statement: everything works in mice, but nothing works in humans. And this prompted us to organize a conference in Washington about 2 years ago. In fact, we published that because of the same issue. The question is, why can't we get good translational work from what works in mice to humans? And in fact, most things don't even work in mice, let alone dogs, cats, rhesus monkeys, baboons, spider monkeys, and so forth. So this is where we have stood till now with regard to central nervous system aging, the dementias, for example. Everything works in mice, nothing works in humans. And from an investor standpoint, Alzheimer's is the graveyard of companies. And the reason is we have tended to fail, not surprising. These are data taken from 9/13, that is this month. And I'll look at the clinicaltrials.gov on interventional trials. What you find is there have been more than 3,000 trials for dementia, many on Alzheimer's and Parkinson's. And by global consensus, none of these trials has demonstrated a change in the disease course, anakinumab notwithstanding. So the question is, can we do better? This is where we stand, for example, with regard to monoclonal antibodies and the dementias, and particularly Alzheimer's. We have one drug, the anakinumab, that the FDA certified in June of this year. We have a number that have been felt to be failures, although some of them are coming back, and we have a number in the pipeline. The problem is that even when I look at the best of these, for example, anakinumab, this is the anakinumab data plotted out, comparing it to placebo. And what you find is that with each increase in dose, with the exception of the third highest dose, you find that there's a slightly better delay in symptoms. But after that, the disease course parallels the underlying course of placebo. That is, you're not getting much of an improvement. You're stepping off, delaying somewhat the disease, but the vector is parallel. That's not what we want. What we want is this. We want to show that we can at least stabilize the disease, if not actually improve the cognitive status in an age-related disease. So the question is, why haven't we done that? And the answer is, we've been digging in the wrong place. And when you're digging in the wrong place, it doesn't matter how big the NIH budget is or the NHS budget is, or the budget, in this case, for Biogen Eisai of more than $1 billion. Because if you're digging in the wrong place, then digging deeper, despite the costs involved and the resources put into it, or how big your excavator is, doesn't matter. If you're digging in the wrong place, you need to find the right place. Beta amyloid, tau tangles, calcium channel blockers, immune dysfunction are not the right place because they have failed. What we need is something a little bit better. So again, to reiterate, when it comes to Alzheimer's as an example of aging and age-related diseases, we know a great deal, but it's not enough of an understanding of it. So back after that same conference, which I summarized, I put out this paper, which was a landmark paper and brought an awful lot of investors to us as cold calls and generated more than 600 reprint requests, as well as invitations for me to chair a number of conferences on Alzheimer's. But what I was really saying was, it's not that we need better technical ability or a number of targets, we need to understand the disease. We need a unified systems model of dementias and age-related neurodegenerative disease. Now, when I say a unified systems model, let me be a little more specific, particularly with regard to unified to start with. When I say unified, what I mean is we know a lot of the upstream risk factors, for example, ApoE4 alleles, or a number of infectious diseases, hypertension, hyperglycemia, trauma, toxins, for example, paraquat and dioxin, that have increased risk factors for neurodegenerative diseases like Alzheimer's and Parkinson's. And we know a lot of the downstream biomarkers, for example, beta amyloid plaque, tau tangles, and so on. But the question is, what's going on between them? If I have two ApoE4 alleles, why, how exactly does that increase my risk of Alzheimer's? And not in everyone. And likewise, if I have an infection or had a history of head trauma, exactly what are the processes that result in downstream clinical neurodegenerative diseases? And looking at this, particularly with regard to the dementias, what you find is that there's a lot of overlap. So, for example, there really is, unfortunately, a syndrome of Alzheimer's disease. And while that's generally felt to be cognitive in function, for example, memory deficits or executive function disorders, you also find that some of these patients have motor function disorders. Likewise, with Parkinson's, was generally felt to be a motor dysfunction, for example, the substantia nigra or minor nucleus problems. Some of these patients also have cognitive disorders. There's a fair amount of overlap. Likewise with frontotemporal dementia. When I look at vascular dementias, many of these patients show vascular dementia findings on autopsy. So it's not as clear as just these are individual. There is a certain amount of gray zone and overlap, all the more so when you look at the full alphabet soup of many of these other age-related neurodegenerative diseases. In fact, many of our patients are atypical. They don't quite fit in any category, or they have mixed categories. They have some of each, as I've said, with regard to Alzheimer's and Parkinson's. So over the past decade or so, more and more people have begun to wonder if there is a unified process, an age-related neurodegenerative process, that many of these clinical syndromes share at some level. And the same is not only true of dementias, it's true of cardiovascular disease, age-related cardiovascular disease. So while many of our patients have CHF or have myocardial infarctions or strokes or capillary pruning peripherally, what you find is that many patients have a little bit of both, or they have some overlap in the way they present. And the same is true when I expand that set of definitions of clinical syndromes. You find patients with mixed disorders or atypical disorders, which don't quite fit into any one category. The question again is why? And again, the suspicion is we're dealing with some underlying processes that are encompassing all of these different syndromes, whether we're looking at central nervous system or cardiovascular disease. And the reason that we have different clinical presentations is because each of us start with a different genetic, epigenetic, and cellular background. Our behavioral histories are a little different. And the result is we have different clinical outcomes, but there's all played out on a unified stage of an underlying process. And that process probably has to do with cell senescence. In this case, what we're seeing is the same thing in central nervous system cells, endothelial cells in the vascular system, chondrocytes in the joints, osteocytes in the bone, renal podocytes, leukocytes peripherally, and skin cells, and other systems as well. So that's the unified portion. Now, what I mean by a systems model is something a little more subtle, but equally critical. And let me give you an engineering example. This is the cockpit of an Airbus 380. In the upper right is a Rolls-Royce Pro 15 jet engine. On the lower right is a Trent 900 turbofan that powers the Rolls-Royce, which powers the Airbus. But if a single fan blade shown here in the foreground fails, a good engineer doesn't simply ask what's wrong with the fan blade, but asks what's wrong with the fan blade in the context of the Trent 900 in the Rolls-Royce Pro 15 jet engine in the Airbus 380 at 35,000 feet, going hundreds of miles an hour at a high RPM and temperature. That is, it's a systems issue, not simply a component issue, but a component within a system. What's going on in the whole system? Now, the same thing is true medically. If I look, for example, at biomarkers, that is signs and symptoms of COVID, no matter how carefully I measure the data on each of these typical signs and symptoms of COVID, that doesn't tell me how COVID works. What I need to understand is the system, that is the COVID virus in the context of the human body and the human immune system. And the same thing is true with regard to aging. These are typical macroscopic biomarkers of aging that we see clinically, and we see in ourselves and those around us. But they're not aging. They are outcomes, biomarkers. They are downstream components, if you will, of aging, but they don't tell us how the system works. And even at the microscopic level, the same thing is true. Knowing that there is a decline in DNA repair, no matter how carefully we measure it, or a declining ATP to reactive oxygen species ratio, methylation, acetylation, telomere shortening by itself doesn't tell us what's going on. These are adequate biomarkers, but they're not the system itself. They're not the process. And we need to go a little more deeply in our thoughts to understand aging and what to do about it. The same thing is true of the dementias. These are typical biomarkers for many of human aging-related dementias that we see. For example, failure of the blood-brain barrier, lymphatic changes, glial cell activation, and so on. But these are not the dementia itself. These are biomarkers of it. What we need to understand is the process. And even at the genetic level, the same thing is true. I know of more than 200 genes that have been implicated in increasing or in some minimal sense decreasing your risk of dementias, but they don't explain the process. And as we'll see when we get to aging, the same thing is true. It is not a genetic disease so much as something that's a little more complicated and subtle than that. This is a quote from a friend of mine that many of you know. This is Dr. Len Hayflick. And 60 years ago this year, he first described cell aging. And he points out something that's critical, which is almost all of us would agree, that the single major risk factor for age-related disease is aging. But most people then ignore aging totally and focus on the histology, the physiology, the biomarkers, the genes, without asking themselves, what is aging doing? And I think we need to understand it, because unless we actually understand the process at the fundamental level, then we can't intervene at any fundamental level. So if we're going to intervene in aging, we need to understand it. Let me take you on a broader perspective of aging than when you're used to. These are a number of species that I think we're all aware of. Human beings, for example, mean lifespan is about 3/4 of a century. That is Coco, the gorilla. I was her babysitter for a year at Stanford, which was a remarkable experience, especially humiliating since she had a bigger sign language vocabulary than I did. But Coco died a few years ago, short of a half a century, which is typical for gorillas. Typical domestic animals live about 15 to 20 years, depending on the individual breed involved in species. And the same is true of the differences with rodents. For example, typical laboratory rodents live about 2 or 3 years, depending on, again, the particular breed and type you're looking at. But the question would be, why do we age at different rates? And most of us would then say, well, it's genetic, which is part of the answer, because it's true that, for example, with that mouse, I share about 75% protein expressing genes, and presumably the secret to aging has to do with the other 25% of genes. But that's not an explanation any more than knowing that 2 ApoE4 alleles increase your risk of Alzheimer's explains why it increases your risk of Alzheimer's. The same is not only true between species, but also within species. For example, this is a group of Hutchinson-Gilford progerics, and 20 years ago, I gathered this group at the White House, where I gave them a tour of the White House, taking them places you're not allowed to go and taking pictures you're not allowed to take. And we had a fine time until Sammy and Ori in the front row on the left there with the red baseball cap and the orange Hawaiian shirt managed to sit on a pressure-sensitive switch up to the president's quarters, and we were escorted out here to the North Portico to take these pictures. Now, these children have an average age at death of about 12.7 years. They die overwhelmingly of age-related disease, typically cardiovascular disease, strokes, and heart attacks, for example. But why? Well, we know that most of these children that we've looked at have a lamin A gene abnormality, and we tend to ascribe it to that, but that's not an explanation. The question is, why does the lamin A gene result in an aging that's at least 4 times faster than most of ours? Why does that occur? Knowing where to look helps, but it doesn't tell us how it works. We need to go a little further. The same is true if we look a little more deeply at the biological context. So, for example, every cell in your body derives from a fertilized ovum, and the ovum came from your mother, that came from her mother, and you could trace that line of germ cells back about 4.5 billion years. Now, retrospectively, that germ cell line never aged, and yet your somatic cells demonstrate age-related changes now in a matter of decades. So you can't say aging always occurs, because sometimes it doesn't, sometimes it does. And nor can you ascribe it to mitochondrial changes, because every mitochondria in your body derives from your mother, who got it from her mother. And that line goes back something short of 2 billion years, depending on who's making the estimate. And when you inherited those mitochondria, they showed no age-related changes. And yet, in most of your cells, there are age-related mitochondrial changes now. So why does aging not occur in mitochondria for almost 2 billion years? And yet, in a matter of a few decades, it plainly occurs in the mitochondria. We'll come back to why that happens in a minute. Well, it is clear then that aging is not entropy. It is entropy versus maintenance. If we're looking at the germ cell line, what you find is that maintenance is essentially flawless for a long period of time. Where if I look at the somatic cells in the 80-year-old man's hand on the left in the wheelchair, whether I'm looking at fibroblasts or vascular endothelial cells, or for that matter, cells in the brain or in the heart, what you're seeing is that there are age-related changes related to a very subtle change in maintenance. It is not entropy per se that causes aging. It's entropy in the face of gradually failing maintenance. In fact, it's sort of like Red Queen theory. This is a quote from Through the Looking Glass by Lewis Carroll, where the Red Queen says you must run as fast as you can just to stay in one place. And that's essentially what our cells are doing as best they can. And that running in one place fails as we get older. And the question is exactly what's going on and can we intervene? Well, if we look more carefully at any molecular pool in your body, whether it's the collagen and elastin between the cells and my face, or whether we're looking at beta amyloid, for example, and outside the neurons in the central nervous system and the effects of microglial aging, or whether we're looking at mitochondrial dysfunction and aerobic enzymes, for example, cytochrome C oxidase, or for that matter, catalase in your cells. No matter what molecular pool we're looking at, we find that turnover is relatively fast in young cells, but as cell senescence progresses, what you find is that the turnover rate goes down. And as a result, you're no longer washing out damage that's signified here by the little red stars as quickly. So you begin to get a gradual accrual of damage over time as the turnover rate slows down, the molecular recycling rate slows down. And that becomes quite significant as we get older, and the result is cellular dysfunction and cell aging. In fact, you can look at this mathematically. This is taken from my Oxford University Press textbook, What it essentially says is that young cells with a relatively rapid rate of molecular turnover have a low accrual rate of damage per unit time at equilibrium compared to older cells with a lower rate of turnover and consequently a higher percentage of accrued damage per unit time. And this is true no matter where in the body we look. But to look as an example of central nervous system aging, for example, dementia, what you see in Alzheimer's patients, and we've measured this, is a lower rate of turnover, for example, of beta amyloid, tau proteins, and mitochondrial molecules. So if we're looking, for example, at beta amyloid as one example, you find that in young cells, the microglia, for example, will produce beta amyloid, secrete beta amyloid, bind the beta amyloid molecules, reintroduce them back to the cell, degrade them. And this is a continual recycling of these molecules. And in young cells, the result is you don't get much in the way of beta amyloid microaggregates, whereas when we get older, the turnover rate slows, you begin to get this gradual accrual of beta amyloid microaggregates as they cross-link. You might ask, what's the effect of some of the different alleles you inherit? So for example, if you have two ApoE2 alleles, which have a consequently low risk of Alzheimer's, the reason is that the molecules you create from the beta amyloid molecules with your ApoE2 alleles are relatively non-sticky. So you don't get a lot of damage even when you have a slower rate of turnover, for example, when you're 40 or 50. However, if you inherit two beta ApoE4 alleles, what you find is you make stickier molecules, and the result is an earlier age at onset of Alzheimer's and a more significant disease course. So whether you have ApoE2 alleles or ApoE4 alleles, whether you make non-sticky or sticky beta amyloid, it all plays out on the stage of cell senescence and the slower rate of turnover of molecules. And the result is that we begin to see as we get older, beta amyloid plaques earlier in those with stickier molecules, for example, ApoE4 alleles, and later, or maybe even not at all, in patients with low risk alleles like ApoE2 alleles. And the result of that is clinical dementia, cell dysfunction, cell death, loss of synaptic neurites. And the same is true whether we're looking at beta amyloid, tau proteins, mitochondrial molecules, which you look at in a minute, inflammatory changes, and so on. And in fact, if we're looking at mitochondria, we see the same thing. About 99% of the aerobic enzymes responsible for mitochondrial function derive from the nucleus. And what we see is there's a lower rate of turnover as cell senescence progresses. The response then is that the oxidative damage that occurs in mitochondria tends to increase over time as the molecular turnover rate slows down. So the result is a lower and lower ratio of ATP to reactive oxygen species with age, and a higher membrane porosity with age, because the lipid molecules of the mitochondrial membranes are also being turned over more slowly. So oxidative damage results in an increased percentage of damaged lipids and therefore leaking a membrane. And likewise, scavengers, for example, superoxide dismutase of either variety or catalase also being turned over more slowly, so scavenging less effective. And likewise, when there is damage, the recycled molecule rate is turned down. So for four independent reasons, we get this exponential increase in cell damage as a result of mitochondrial dysfunction, solely secondary to cell senescence processes occurring within the nucleus. The same is true of DNA repair. If we look at the four families of DNA repair enzymes, all four independently show slight changes, subtle changes, but slight slowing of the rate of recycling. And the result is an exponential increase in the rate of mutation, not per time, but per lifespan. That is, it's not a matter of whether you're a 2-year-old rat or an 80-year-old human. It's not the years, it's the rate of cell senescence that results in this change and this increase in mutation and therefore cancer rate, which again goes up exponentially over lifespan, not per years. So the overall process is that of cell aging. And upstream, we have a lot of risk factors that have an impact on the rate of cell aging. For example, exposure to toxins, like, as I say, paraquat or dioxin, traumatic head injuries or traumatic injuries to your knees or other part of your body increase the rate of cell aging. Likewise, vascular effects like hypertension, hyperglycemia, and likewise infections will increase the rate of cell aging in various cell populations. And that causes cell loss, which increases cell division, which causes telomere shortening, which actually modulates the pattern of gene expression, which slows molecular turnover. And the result is cell failure, dysfunction, tissue failure, organ failure, and downstream effects that are listed here as just some examples. But the question is, what can we do about any of this? So we know that we've been trying to deal with upstream risk factors, for example, mitigating the effects of hypertension or making sure you're euglycemic minimizing head trauma and other trauma to the body, dealing with microbial disease, and those have an effect. But they don't stop the effects of, for example, cardiovascular aging. They simply slow it if I can control your blood pressure and your glucose. Likewise, downstream, we've been trying to deal with, for example, beta amyloid plaques in the central nervous system using monoclonal antibodies, but again, with minimal effect. So can we intervene more effectively? The answer is yes. We can actually directly have an impact on cell aging. It's been done and it works. Here's an example. This is a paper from Science, the journal Science, from Andrea Bodnar and her group out in California some 20 years ago. And what she showed is if you look at the epigenetic pattern of young cells, very individual, older cells, in this case, human fibroblasts, very individual, and if you use telomerase to reset that pattern and reset telomere length in old human fibroblasts, you get young human fibroblasts, morphologically, histologically, physiologically, and in terms of the pattern of gene expression as diagrammed here. And the same is true when we look at tissues. So this is a paper from Walter Funk and this group, again, out in California some 21 years ago. And what they did was show that you can take young human skin and you get a very distinctive pattern in terms of the histology. If you look at old human skin, you get a very distinct pattern in terms of the histology. But if you take old human skin cells and telomerize them, you end up growing what looks like young human skin. So we know that we can reset aging in human cells. We know we can reset aging in human tissues, not only in skin, but the same is true of human osteocytes and bone and vascular endothelial cells. So for example, literally you can take vascular endothelial tissue from the coronary artery of an old person and grow what appears to be young vascular tissue. We know we can reverse aging in cells and in tissues. The question is, can we do that in organisms? Well, that was at least partly accomplished about 10 years ago at Harvard with Rhonda Pinot's group, but he used a method where he altered the germ cell line. It was accomplished in a much more effective manner by Maria Blasco's group in Madrid, here in this case, some 9 years ago. And what she did was she used telomerase gene, in this case mTERT, in an AV9 vector to deliver this to adult mice and show that you could not only improve the physiologic measures like bone mineral density, subcutaneous fat, immune function, and a number of other parameters, but also behavioral function. That is, these animals acted more like young animals, as did the case from Harvard with Rhonda Pinot's group, and the lifespan increases. So we know we can at least partially reset aging in the organism, not simply in cells, not simply in tissues, but in organisms. The question is, can we do that in human beings? Which is what we're about to do, and it's what excites me. We're going to concentrate, obviously, on efficacy and safety, but credibility is also key. We're about to do something that I suggest is never been accomplished in human history before, which is to show that we can fundamentally reverse aging at the genetic, epigenetic, and cellular levels, and in so doing show that we can cure and prevent a number of age-related diseases. As a result, we've been able to attract a remarkable scientific advisory board, including, for example, Suzanne Hendricks, who actually helped design that Biogen Eastside study in aducanumab, Kurt Whittemore, who was a postdoc who helped do that study in Madrid with Maria Blasco, showing you can reverse aging in organisms. Joe Arahu, who does many of the aging studies in dogs worldwide for Alzheimer's and other dementia studies. Ramon Azouz, who runs an institute devoted to translational gene therapy work in neurology. Likewise, our clinical advisory board. These were people who were chosen for global credibility and for their skepticism. We wanted people who saw the value in this model and want to see it tested rigorously and correctly. So we've got people who are quite clear that we're going to do this right. We will be starting our human trial with central nervous system disease, specifically the dementias and more specifically Alzheimer's. We're moving on then to cardiovascular diseases, plural. In fact, I have a review paper that will be coming out next year on exactly that issue. Moving on then to osteoarthritis, osteoporosis, chronic renal failure, immune senescence, dermal aging, and so forth. This is a platform therapy. We will be using this for all age-related human diseases, starting with central nervous system neurodegenerative diseases and moving on from there. We think that we can alter aging at the most fundamental level because the data support it. We know we can alter aging at the fundamental level in human cells, in human tissues, and in a number of organisms. So the answer clearly is yes. The question is, can we do this to show that we can cure and prevent age-related diseases? And the answer is, we almost certainly can, but we'll have to see what the human trials show over the next few years. Thank you very much for joining me. And should you have any questions, please feel free to contact me. And I hope you enjoy the conference and find the material interesting. Thank you again.